Dear students, Assalamu alaikum. Today I am going to deliver a lesson for class 11th, subject zoology. Since we are studying the structure of an eukaryotic cell, and in our last topic we have discussed about the structure of cell wall and cell membrane of an eukaryotic cell. Now today we will discuss about a very important point of the eukaryotic cell, that is the endomembrane system. Endomembrane system of eukaryotic cell is basically a group of membranous organelles that work in coordination to modify, package and transport lipids and proteins. Now these membranous structures that are involved in endomembrane system are endoplasmic reticulum, Golgi complex, lysosomes and vacuoles. One thing you must remember here, that the mitochondria, chloroplasts and peroxisomes are also membranous organelles, but they are not considered in endomembrane system because they are not coordinated with this system. As such, they are not included in endomembrane system, and thus the endomembrane system consists of 4 components, that is endoplasmic reticulum, abbreviated as ER, Golgi complex, lysosomes and vacuoles, as is depicted in this diagram. Here this is the nucleus of the cell, of an eukaryotic cell. Then this nucleus is attached to this endoplasmic reticulum, and endoplasmic reticulum then communicates with this Golgi complex, and then they are transported, the components formed, the lipids or proteins which are packaged in these ribosomes are transported through vesicles to the plasma membrane through transport vesicles. Now endoplasmic reticulum is a network of tiny tabular structures, as you can see in this sketch, which are scattered in the cytoplasm around the nucleus. This is the nucleus. This endoplasmic reticulum divides the intercellular space, actually intercellular space of the cell, into 2 components. Now, the first component is the luminal component. That means the component that is inner to the endoplasmic reticulum. This one. This is the luminal compartment. And the second compartment is the extraluminal compartment, that is outer to the endoplasmic reticulum. This is the extraluminal compartment. Now this lumen of this endoplasmic reticulum contains is filled with a fluid and contains many enzymes and other proteins. Basically, there are 2 types of endoplasmic reticulum occurring in cells, that is RER, which abbreviates as the rough endoplasmic reticulum, and SCR, that stands for smooth endoplasmic reticulum. Now often a question is asked during your examinations to point out the main differences between the rough endoplasmic reticulum and smooth endoplasmic reticulum. And in this connection, you must remember that rough endoplasmic reticulum has ribosomes on its outer surface, as is shown here. These dot-like structures, these are the ribosomes present on the outer surface. Now this endoplasmic reticulum is present more in such cells which secrete proteins and enzymes. This rough endoplasmic reticulum is also involved in the synthesis and transport of proteins. Now, the smooth endoplasmic reticulum. As the name suggests, smooth, that means it has no ribosomes attached to it. That is, its surface is smooth, because ribosomes are not attached to its surface. Now, it is present more in those cells that synthesize lipids, and this smooth endoplasmic reticulum is involved in the synthesis of lipids and steroid hormones. Now, this was all about the endoplasmic reticulum. Now, next component of the endomembrane system is the Golgi bodies or Golgi complex. These Golgi bodies were first observed by a scientist known as Camillo Golgi in 1898. These Golgi bodies consist of many flat, or you can see the disc-shaped sacs, which are also called as cisternae. And these cisternae are stacked parallel to one another in concentric manner near the nucleus. Now, these cisternae have a distinct convex or cis face. This side is the cis face. Cis face pertains to the forming phase, where the substances are being synthesized or formed, facing the nucleus, and a concave phase, also known as the trans phase, or also known as maturing phase, that faces the outer surface of the cell. This is the trans or maturing phase. Now, after Golgi complex, the another component of endomembrane system is what we call as lysosomes. Now, these spherical or irregularly shaped vesicles are in fact the lysosomes. These lysosomes are thus spherical or irregular, and they are single membrane bound vesicles. You must remember this. These are the single membrane bound vesicles formed by the process of packaging in Golgi cisternae. These lysosomal vesicles, they contain a number of hydrolase enzymes like lipases, proteases, carbohydrases, etc. And these enzymes optimally work at an acidic pH. After this lysosomes, the fourth and the last component of this endomembrane system are the vacuoles. Now, vacuoles are membrane bound spaces, in fact, which are present in the cytoplasm. The single membrane boundary of the vacuoles is called the tonoplast. This question is often asked in multiple type questions. You must remember, therefore, tonoplast is the single membrane that covers the vacuoles. The tonoplast of plant cell facilitates the transport of a number of ions and other molecules into the vacuole against the concentration gradient, and so their concentration is significantly higher in vacuoles than in cytoplasm. Vacuoles are of three types. There are three types. Number first is the sap vacuoles. Sap vacuoles are the vacuoles of plant cells. Sap vacuoles are the vacuoles of plant cells, and that contain, in fact, this water, minerals, and excretory products, etc. Second type of vacuole is the contractile vacuoles. These vacuoles are found in unicellular organisms like you must have heard about amoeba, paramecium. They contain the contractile vacuoles, that help in excretion and osmoregulation in these animals. The third type of vacuole which is present in the eukaryotic cells is the food vacuole. The food vacuoles are also common in organisms like amoeba, paramecium. They are formed by the engulfing the food material by phagocytosis, and digestion occurs within them. Now, this was all about the endomembrane system of eukaryotic cell. Now, in our next topic, we will study about the other cell organelles like mitochondria, chloroplasts, etc. Until then, goodbye and good luck.